Okay, this is the introduction to mitosis. This is going to be 5 short screencasts. And what I'm going to do with this one, I'm going to keep adding these screencasts up, pretty fast here. So this way we can get mitosis done. All right. Listen, learn, draw. All right. This is now going back. So before we start this is chapter 5 in your book, Cell Division. Some great pictures in chapter 5 about the cell cycle and mitosis, and I'll refer to them back. But for now, see you on the other side of the animation.
Is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between 2 daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into 2 daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
Okay, so this was the first one there. And the reason why I wanted to show you this one, because now you have 2. You have one of an animal cell and one of a plant cell. All right, so we go back to the PowerPoint for just a couple of minutes here. All right, there you go. Just want to give you the vocabulary and go over the important few words that you might have heard in the screencast. All right, number 1, centrioles. Remember that from way back earlier in the year. Those are the 2 cylindrical barrel-shaped structures found near the nucleus involved in mitosis. Centrioles are contained within a region called the mitotic center, or the centrosome region. All right, so the centrioles and the centrosome, and I'm going to add that to the notes right here. So when they go up online, you'll see centrosome in there. All right, so centrosome, centrioles in it, and don't forget the centrioles are only in an animal cell, which is why you didn't see them. But there is a centrosome in a plant cell. All right, an aster. Okay, an aster is just the bunch of microtubules that radiate out from the centrosome in both a plant and an animal cell. Okay, you can see the asters right here. Let me get rid of this, hide that, sorry. I'm not stopping it. Okay, so an aster. And of course, spindle fibers. Spindle, okay, is what actually attaches to chromosomes, all right, which brings about an orderly distribution of chromosomes into 2 new cells. All right, so those are the most important words, along with centrosome, which will be part of this. So each one now, you should be labeling these as well as DNA in each one that you draw. All right.